So, good day everyone. I am NJP Salaga, currently a BS Biology student of Iloilo Doctors College. So, something really fascinating to me is that every cell in your body has the same DNA in it. If the protein that makes each of your cell type different from each other. That's why your hair is not the same as your eyes and as your nose. So, proteins are long molecules that have a specific three-dimensional shape. That what proteins can do is a lot. They can detect signals, they can be signals, they can regulate your body temperature, they can help your cells recognize the nutrients that they need. But what is really critical is they have to maintain that three-dimensional shape. It is a really specific structure, and if they are experienced condition outside of their normal range, they will denature or they will fall apart. So today, what we're going to do is we will break proteins. Or this experiment entitled as protein denaturation. So what are we going to use? So this is the materials. So first we're going to have 4 mini bowls for our proteins. Next is 4 glasses for our conditions, a scissor, a fork, a tape, of course an alcohol, a tap water, and for our heating element we're going to use a heater. So for our proteins we're going to use an egg. So let's begin our experiment. So first let's prepare the 4 glasses for our 4 condition. The first condition is the albumin or the egg white without any substance added. So in the second glass, where the second condition we're going to put the 75 milliliters of tap water at room temperature. And next, for our third glass, where the third condition, we're going to put also a 75 milliliters of an alcohol. So lastly, in our next condition, we're going to add the hot water. So it's already heated, and also it is a 75 milliliters of hot water. So next, we're going to cut the eggs and get the egg white. So we're going to use 2 eggs for our conditions. So let's begin. So we're going to separate the egg white from the egg yolks because we are going to use only the egg whites because that is where the albumin or the protein can be found in the egg. So this is our albumin, or we're going to use the egg white for our protein. Before we're going to proceed in our major experiment, we're going to label all the conditions. If you're done labeling your glass with your conditions, you're going to add the egg whites or albumin. So this is the result of our experiment. So this is the protein in different type of condition. This is the protein in the hot water, in the alcohol, in the tap water, and this is albumin or an egg white without any substance added. So this is it. So we have our 4 different conditions now, and let's think about what is going on in them. In our control, we have not added anything to our egg white, and that means the albumin or the proteins are still tightly bound up in their specific 3-dimensional structure. Unlike on our 3 conditions here, which is the tap water, an alcohol, and the hot water, what happens, however, when they get denatured is that 3-dimensional structures falls apart. Proteins, and they take up more space, and they actually become this kind of white, milky, long strand of protein. So to sum up, we've denatured proteins different ways today, but proteins get denatured every single day. It's every kind of cell needs proteins in that 3-dimensional shape to survive. So your broccoli have proteins, your carrots have proteins in them, and when you cook them, you're affecting the shape of the protein. That's why it changes texture, that's why it changes color. So while you're going through your day, if you're eating your breakfast, your lunch, your dinner, your fourth meal, whatever it is you are having, think about the ways that the proteins have been denatured or the ways that they will be denatured. Just like our experiment today, called protein denaturation. Once again, thank you for breaking things with me today, and I hope you'll learn a lot from me today about protein denaturation.